Okay, so this is our presentation on Lake Hunter biodiversity.
Lake Hunter is located in Lakeland, Florida, and borders the small but busy Highway 563. It's got development on both sides with heavy vehicle and foot traffic.
So these are the two hypotheses that we use in our study. The first one is our null hypothesis and suggests that all lakes in Lakeland are the same considering their water quality. We suggested for our alternative hypothesis that our lake, Lake Hunter, will have a lower Shannon index and a lower percent EPT because it directly borders the highway and is heavily impacted by human interaction, such as pollution, fishing, and boat disturbances.
So for the demographics, Lake Hunter is part of the Hillsborough River watershed and drains into the Hillsborough River. It has a surface area of around 93 acres as well as a maximum depth of 9 feet.
The average pH of Lake Hunter is a 9.21, which is incredibly basic. It has a dissolved oxygen content of 10 to 12 milligrams per liter. Its Secchi depth is 1.1 foot. Its chlorophyll A content is 110 to 130. Its nitrogen levels are 2,700 and phosphorus is 265. The water quality levels were generated from the Polk County Water Atlas, with the last collection and recording being in October 2019. The nitrogen and phosphorus levels are dramatically high, as is the chlorophyll A.
So here's some common fish found in Lake Hunter. This is recorded from various fishermen. We actually saw some fishermen out whenever we were sampling. There is an accessible boat ramp, and when we sampled, there were people out there. Common catches include bowfins, sunfish, bluegill, and bullhead catfish.
And then according to the Polk County Water Atlas website, Lake Hunter is considered to be hypereutrophic or poor water quality. There is no obvious limiting nutrient present within the lake, but what is strange is about the lake is the update that the turbidity levels are relatively low when we compare it to historic range. But even considering that, the lake is extremely eutrophic. Some characteristics of a eutrophic lake are high nitrogen, phosphorus, and chlorophyll A levels, which is shown in the recent data collection.
Because there are no strong limiting nutrients, there's an abundance of vegetative growth at most of the sites that we sampled. Some of the represented species are hydrilla, eelgrass, arrowhead, bald cypress, pickerel weed, and Potamogeton pectinatus. All three of these live in different water ranges, either emerging or submerged, and represent the high chlorophyll A levels in the lake.
So the most prevalent vegetation in the sampled areas was hydrilla, and it was seen in 7 out of the 8 sites, as you can see in the chart. This is problematic as hydrilla is aggressively invasive and it crowds the water column, which can negatively impact water quality, and that chokes out the native species. The second most abundant plant was pickerel weed, and it was found in 6 out of the 8 sites. And it grows out of the water and can act as a home for the larger invertebrates. The least abundant plants were arrowhead and Potamogeton pectinatus. Arrowhead is native and it emerges from the water and acts as an indicator of ecosystem health, so the lack of them is concerning. The second is a native grass that lives underwater. It grows mainly in less than 6 inches of water, which we saw a lot of, and its lack may indicate less substrate for the macroinvertebrates to live on.
So the number of different macroinvertebrates that can be observed in Lake Hunter is fairly low, but nonetheless, you can see grass shrimp, freshwater snails, scud, ramshorns, and skimmers frequently within Lake Hunter.
So this pie chart shows the number of species and compares their overall abundance. There were a total of 411 organisms within 15 different taxa that we collected, and almost a third of them were grass shrimp, and the top 5 species made more than 85% of the total. The other 15% was made of the other 10 organisms, which were relatively sparse, and we saw a lot of insect species as well as different indicators.
So looking at how Lake Hunter compares to other lakes, the most abundant, or the highest diversity index was found at Lake John, and Northside Lake Hollingsworth had the highest number of taxa. Lake Morton had the least number of vegetative and macroinvertebrate species, but it also only had a slightly larger Shannon index than our lake, Lake Hunter. So ours was at the lower end of the lakes.
So when we compared Lake Hunter to the other lakes in terms of the percent EPT, we did see the highest percent EPT, which indicated that we observed the highest amount of pollution-sensitive taxa, which was surprising to us considering the amount of human interaction that is present at the lake.
So when we compare Lake Hunter with other lakes in the Lakeland region, it had a lower Shannon index, and this is likely due to the proximity that the lake has to human development, which will be discussed more in our next slide. We were able to reject our null hypothesis that all lakes in Lakeland were the same, as our class data tables show information from all 6 lakes with very different results. Lake Morton, the lake with the lowest biodiversity, had only a slightly lower index than that of Lake Hunter. However, we can't accept the alternative hypothesis because Lake Hunter had low biodiversity, but actually had the highest percentage of EPT out of all of the locations. This made this habitat very complex as the water quality is seemingly poor, as is the biodiversity, and yet the pollution-sensitive organisms are still present. Some justification for this confusion is that only 3 of our 411 species, which is less than 0.5%, were from the ephemera taxa. So there's a chance that some of the commonly known pollution-sensitive individuals may be evolving to live there.
So these are some pictures that we found, or that we took while we were taking our samples. So before the collection of data, we assumed that there would be a lower Shannon index because of the proximity to the lake and human development, such as the major highway and some of the drainage that was depositing waste directly into the lake, as seen in the picture on the left. Considering the level of pollution that was around the shoreline and in the water, compared to that of the vegetation, it was trying to get a true representation of species in the lake, and it was very challenging because of all this extra stuff. We also had trouble finding a safe and helpful entry point to the lake to collect the specimen. Both of these challenges could lead to species that were not accounted for, which meant our Shannon index calculation could have been invalid, but also might have been even lower if we did get these samples.
So some next steps to take when conducting sampling at Lake Hunter, trying to find a better way to get into the water. We found that it was really hard just because of the amount of vegetation, pollution, as well as there were a couple spots where the ground was a little unsteady. So trying to find an alternative way, whether that be bringing more equipment, such as waders, to get farther into the water to make sure that we're getting an accurate sample collection, or even if you use a boat from the boat ramp to get closer to the shore so that you are able to avoid the vegetation. All of those things would be beneficial in terms of trying to find a way that would account for all the species present. And here's just a fun video when we encountered an alligator on the shoreline of Lake Hunter.
We're gonna slow it down. Thank you.
Thanks.
Bye.